Welcome back for another deep dive. Given your background in plant science, I think you'll find today's topic particularly interesting.
Oh, is that right?
Yeah, we're going to be looking at these two research papers all about predicting tomato yield. And trust me, some of the findings are pretty surprising. Well, it might seem straightforward at first, right? Can we tell how much a tomato plant's going to yield just by looking at some simple traits early on? But these studies, they actually go beyond basic genetics. They get into these dynamic physiological measurements. It's really cool.
I see. So, like how the plant's actually functioning, not just its genes.
Exactly. And it got me thinking, as plant scientists, we all know predicting crop yields, that can be a real challenge out in the field.
Oh, absolutely. Especially with all the environmental variables.
So many things can impact those yields.
Exactly. Exactly.
And, traditional approaches, they often focus on the genotype, the environment. But sometimes those methods, they can be a little limiting, especially when we're trying to make predictions early on.
Yeah, you need more real-time data to get a better picture.
And that's what's so cool about these studies. The researchers, they took a different approach.
It is fascinating how they're going, beyond those kind of traditional methods. And now they're using this continuous monitoring
Right.
to really unravel all these complexities of plant behavior.
Yeah, because, if you think about it, usually you would just measure like a plant size or its leaf area at one point in time.
Right.
But, that's not really giving you the full picture.
You got it.
It's like trying to understand how well a marathon runner will do just from looking at like a single snapshot. These papers you sent are really all about like capturing the whole race.
Exactly. They're dynamic systems, and they're constantly responding to everything that's going on around them.
Right.
And one of the studies uses this really amazing technology called Plant-Array.
Yep.
And it allows the researchers to continuously monitor all these different physiological traits of the plants.
Right. And one of the most fascinating findings, it actually revolves around something I bet we're all familiar with, stomatal conductance. But here's the kicker. They found that measuring GRAC in the morning, like that is incredibly predictive of a plant's final yield. They found correlations as high as 0.9 between morning GRAC and total yield. It makes you wonder why we always fixate on those midday measurements.
So what makes those morning readings so special?
Well, think about the conditions during those early hours, right?
Okay.
You've got high light intensity, perfect for photosynthesis. But then you've also got a lower vapor pressure deficit, so the air's not as dry. And that lets the plant take in a bunch of carbon dioxide for growth, but without losing a ton of water through transpiration.
So it's all about striking that balance.
Yeah. High CO2 uptake, but minimal water loss.
Like a golden hour for the plants, maximizing their efficiency.
Exactly. And it really challenges that conventional wisdom, the idea that midday measurements are the be-all and end-all.
Interesting.
They also found something fascinating about another seemingly basic measurement, cumulative transpiration.
Okay, CT. The total amount of water transpired by the plant.
Right. And, it seems simple enough.
Yeah, you just measure how much water the plant's losing.
But they found that under well-irrigated conditions, there was a really strong positive correlation between CT and both total yield and plant weight.
So more transpiration, more growth, more yield seems pretty logical.
On the surface, absolutely.
But knowing plants, I bet there's a twist somewhere.
You know it. Under drought conditions, that relationship flips completely. CT becomes negatively correlated with yield. It seems counterintuitive, right?
So is high transpiration a bad thing when water is scarce?
Not necessarily. We've got to remember plants are incredibly adaptable.
Right. Masters of survival.
Exactly. So when they're faced with drought stress, they basically switch gears. They go from this growth-focused productive mode
to a water-conserving survival mode. Makes sense.
It's like they're saying, hold on, I got to conserve water to make it through this.
Well, what happens when the drought's over? Do those plants bounce back?
That's the critical question, right? And the researchers, they found that during that recovery period, after the drought stress is gone, the correlation between CT and yield, it becomes positive again.
So those plants that can recover quickly and start transpiring again, they're the ones that end up yielding more.
It's all about resilience.
So it's not just about surviving the drought. It's about how effectively they can recover and get back on track.
It reminds me of those time-lapse videos where you see a plant moving in response to light.
I've seen those.
It's like we're seeing a similar response here, but instead of light, it's all about water stress and recovery. And by continuously monitoring these processes, we can really get a deeper understanding of what the plant's going through and how that ultimately impacts its yield.
It's like we're getting a glimpse into the plant's diary.
Seeing its struggles, its triumphs, as it navigates these environmental challenges.
That's a poetic way to put it.
But before we get too carried away with the drama,
Right.
There's another layer to this research that takes us deep into the world of plant anatomy.
Oh, I'm intrigued. Tell me more.
So the researchers, they went beyond just DSC and CT and decided to take a closer look at the stomata themselves. They started exploring things like the ratio of stomata on the abaxial, the bottom,
Mhm.
versus the adaxial, the top surface of the leaves. It's incredible how plants evolved all these different strategies for managing their resources.
A true testament to the power of evolution. And what the researchers found was that these little variations in the stomata, they can have a big impact on how efficiently the plant uses water and ultimately how much it yields. But let's get back to those mutations we talked about earlier.
Oh yeah, the ones affecting those tomato lines.
Right. The researchers used these tomato lines with known yield differences, and some of them had these mutations in carotenoid biosynthesis.
Okay, and carotenoids, those are the pigments that give tomatoes their color, right?
Exactly. But here's the thing. These mutations, they also affect abscisic acid synthesis in the roots.
Wait, ABA, that's the stress hormone, right?
You got it. ABA plays a huge role in how plants deal with drought and other tough conditions.
So what's the connection between carotenoids and ABA?
Well, it turns out that the mutations affecting carotenoid production, they indirectly mess with ABA synthesis in the roots.
Interesting. So it's like a domino effect, starting with the pigments and ending up with the stress hormone.
Exactly. And here's where it gets really interesting. By impacting ADA synthesis in the roots, these mutations indirectly influence stomatal behavior in the leaves.
Whoa, hold on. So something happening way down in the roots is affecting how the stomato is behaving in the leaves.
You got it. It's all connected. And that's what's so fascinating about this concept of reverse phenomics. You start with a desirable trait like high yield or drought tolerance, and then you work backwards to understand the genes and physiological mechanisms behind it.
Ah, I see. So you're kind of reversing the usual process. Instead of going from gene to trait, you're going from trait to gene.
Exactly. And in this case, they started with tomato lines that already showed those different yields, and then they dug deeper to figure out what was happening at the molecular and physiological level.
That's clever.
And it emphasizes how important it is to connect basic plant science with real-world applications.
For sure. By understanding these fundamental processes, we can develop tools and strategies to tackle those big challenges facing agriculture.
Exactly. We've covered a lot of ground here. Maybe we should recap the key takeaways for our listeners.
Sounds good. I think the biggest takeaway is the potential of these dynamic physiological measurements for predicting yield.
Agreed. It's not just about the genes, it's about understanding how the plant is functioning over time. This research just highlights how important it is to have plant physiologists, breeders, and engineers all working together.
Yeah, to develop these next-gen tools and put these findings to work.
It's a great example of how amazing discoveries can happen when different fields come together.
Well said. I think that's a perfect note to wrap up this deep dive on tomato yield prediction.
It's been a great conversation.
I hope you've enjoyed the journey as much as we have.
Until next time, happy researching.